Now we move to Dr. Atul Saraf from Marathwada Institute of Technology, Aurangabad. He will present us the microreactor systems for processing of sugarcane juice. Over to you. Your 8 minutes starts now.
My, my project title is Development of a Microreactor System for Processing of the Sugarcane Juice. So this is the funding details given by the Rajiv Gandhi Science and Technological Commission. So I am working on a microreactor technology. This is the first image shows the conventional reactors in which the reagents are loaded in the reactor. After completion of the reaction, the products are collected from the batch reactors. This is the second image shows the microfluidic channels or microreactors in which the reagents are loaded from one side of the microfluidic channel and products are collected from second side of the microfluidic channel. This is the difference between a batch reactor and the conventional microfluidic reactors. So this is the scale up mechanism for a microreactor system. In conventional reactors, to scale up the reactors, the size of the reactor is increased, but in microreactors, instead of increasing the microchannel size, we are increasing the number of the microchannels. So this is the scale up mechanism behind the microfluidic channels. So origin of the problem, India is the largest consumer and the second largest producer of the sugar products, and the shortage of manpower is the biggest problem in jaggery industry. So because of which many companies closes their jaggery unit. So this is the traditional method to manufacture the jaggery. These are the objectives of our project. The main challenge in the microfluidic channel is to pass a semi-solid liquid, a semi-solid material and sticky material through the microchannels. So in this project we have overcome this problem and we have designed and developed the microreactor system for the jaggery production. Also we have separated water from sugar cane juice, and we have checked the third objective is to check the microreactor system stability and the performance. The current status of the project, we have started with the single microchannel system. So these are the different microchannel system which we have manufactured for a testing purpose. What is the this is the first microreactor, single channel microreactor system, and this is the last microchannel single microchannel system. The significant difference between the first and last microchannel system is we have improved the heat transfer rate, heat transfer area, improved the design, improved the operating pressure, temperature range, cooling and heating arrangement. So to pass the semi-solid liquid from the microfluidic channels, we require a pumping system. So we have developed a peristatic pump for, and these are the few parts of the peristatic pumps. So this is the journey of a peristatic pump. This is the single channel peristatic pump, and this is the last one is the multiple channel peristatic pump. From first to last, we have changed the design quality, improved the flow rate, designed the system for multiple channels, and we have controlled the improved the control system. So this is the first single channel system in which it is attached with the water recovery system. Water is collected here, and here we are collecting the jaggery. This is the same second video shows the same system but without water recovery system. The temperature for this system is around 167 degrees Celsius. This is the temperature, this is the peristatic pump, and this is the sugarcane juice. In next step, we have developed two prototypes of 50 kg capacity. This is the first prototype and this is the second prototype. We have selected, depending upon the performance, we have selected the second prototype for the numbering of purpose. So this pilot plant consists of a peristatic pump, microreactor system, water recovery system and control panel. These are the different samples of jaggery produced by this system, and this is the present pilot plant of a microreactor system. The capacity of this plant is 2 TCD, and residence time, that is the time required to convert a sugarcane juice into a jaggery, is 30 seconds. This is the biggest advantage of this system. And presently we are working on the energy optimization and mainly on energy optimization. We have also developed few more micro reactors for chemical and pharma industries. The first is micro reactor is alternating magnetic field micro reactor. Second one is electrochemical micro reactor. Third one is a photoflow reactor, and fourth one is a microwave micro reactor.
Thank you so much. Looking at the potential, do you already have some industry contacts or industry which has shown interest?
Presently we are working with few sugar industries. Means we are taking a juice from a sugar industry and we are processing, use it for the jaggery production. They are ready for taking the products.
In the rural areas where jaggery is made, this system that you are proposing, which is electricity dependent, do you see any constraint?
Yes, sir. We are working on the energy calculation, but we are proposing alternating source for electricity. Means indirect heating is the alternative source for this machine.
Indirect heating using combustion
Steam or yes.
Okay.
So is your product the machine by itself? A tabletop machine that
Yes,
I can
tabletop
buy
machine.
on and put it in my kitchen?
Yes, it is possible.
Or is it, and even if, how does it compare in terms of output compared to, the traditional way of doing it?
Commercial products require or the conventional methods require the skilled labor or skilled manpower, but it is a completely tabletop model, and if you set the temperature you get the desired output. Means these are the samples at different temperatures.
So the USP is fresh jaggery in
Yes,
your home.
and the time required for manufacturing of jaggery is 30 seconds.
Can I just add a comment to where it would be a good idea to do a compositional analysis check of the two products? A compositional analysis check of what jaggery produced by your machine and what is available, just to have a head-to-head comparison. This is what I feel.
Ultimately, you will have to justify the cost of production.
Yes.
Ultimately.
I think the point really is what is the USP for, why should somebody invest into buying a micro reactor based technology than what is conventional. The labor cost is one thing, that's all you are harping, but I think eventually the product quality, as ma'am is mentioning and sir is mentioning, is there a differentiation there compared to what is conventional?
No, we can produce same jaggery with the help of this machine.
I think you may want to see whether there is actually an improvement in the quality.
Okay.
That would probably be the most attractive part of this. Right? I mean, there are today small scale jaggery making plants that are used today, right?
Sir, that is why they have been invited for the training program at IIT Bombay.
Right. No, so is there a pain point for those plants? Is labor the only pain point, or is there something else?
टाइम सर 14-15 जॅगरी युनिट चालतात शेतकऱ्यांची
हा.
आता ह्यांचं टाइमच इतका रिड्यूस होतोय.
No, but is that time a pain point? People
Yes.
may be okay with 14-15 hours.
Manpower is the-
that is we sometimes we perceive that as a pain point, but is it really the pain point? Is needs to be checked.
Yes, manpower is.
It could be. I'm not saying that it is not, but there has to be a
Yes.
because there is a capex investment, right? Labor is an opex investment. This is a capex investment. Is there a clear USP on something? And ultimately, it is always the quality. Is there a nutritional value? Because I understand that heat transfer is what you are harping at, right? The area to volume ratio is what micro reactors offer, right?
Yes.
So then there has to be a advantage of that in terms of the quality of the product. You get consistency of the product. It could be color, it could be nutritional value, it could be many things, right?
I think the time what you are telling is mostly dependent on temperature.
Temperature and heat transfer area.
Yeah.
That is surface area to
Again,
volume
that
ratio.
temperature becomes a big cost.
And what is TCD? I'm sorry.
Major motivation, because for a farmer, it doesn't make a sense meter square per meter cube or the time
Yes.
as he was mentioning. He has I mean, possibly your time, the cost of time is not there. So what would be the major motivation for a farmer to shift from a conventional bagasse-based heating system to micro-reactor system?
No, second thing is, what about the availability of juice? So every day if you want a jaggery, every day you have to bring some juice and start producing jaggery? Or, and if it is tabletop, then you are producing only when you need. So overall utilization of the machine itself is very short term. So in a way, I think the logistically, people will say, I will buy commercially instead of investing in such a machine. When you have to have the raw material every day acquired and then you produce the jaggery, something like that. So is it operationally tedious?
रॉ मटेरियल इन्स्टेड ऑफ साखर कारखान्यात घालण्यापेक्षा यांच्याकडे जास्त भाव मिळतो म्हणून जॅगरी युनिट जे छोटे छोटे त्याला जातात ते प्रत्येक शेतकरी आणून टाकतो
Tabletop, so it could be a commercial unit where you keep manufacturing jaggery and sell it again. Right?
Sir, this is the tabletop, but this is the presently this is a 2-ton capacity plant is there.
No, but the question.
Tabletop not for the home users.
No, sir.
That people are thinking it is home users.
It's a 2-tonner machine, that's what he says.
Converting it to various forms. If the jaggery is available either as the block or
Sir.
pocket.
टेबलटॉप झालं तर तुमचा प्रोजेक्ट
You make it into different forms.
So it is possible. We have to control the flow rate. And
सर
we
व्हॉट
can
इज टीसीडी
manage that.
So maybe those kinds of new things that has to be brought in. And the market can completely be different.
Do you already have competition there?
Professor Tikekar thought perhaps was like sitting in Sahkar Nagar in Pune. In his house, he can sort of get juice every day from the, and then sort of make your fresh jaggery or something. Is this what is the proposition?
No, sir.
So what is the
It's
proposition?
a small manufacturing unit. Rather.
Units. Yes, sir.
So for farmer, this is
Farmer.
a technology developed
For the farmer.
keeping end user as the converter of juice to the jaggery. The farmer is your client.
Target. Yes.
So then can you add a solar cell on top and
It is possible.
solar panel, whatever?
Are the colors market friendly? You showed 4 different colors pans,
Yes.
वेगवेगळे. Are they market friendly? मग आम्हाला गुळ बघायची सवय म्हणजे ते लाल ह्याच्यातच आहे. तो जर प्रोडक्ट तुम्ही ह्याच्यात ठेवला तर ते म्हणणार हे काहीतरी वेगळं दिसतं
टेस्ट
आपल्याकडे.
वरून सुद्धा लक्षात येईल. ऑटोमॅटिक चेंज होतो.
No, but what sir says is right की if गुळाचा रंग जो आहे तो मला पर्सनली सोनेरी किंवा समथिंग लाईक दॅट इज मोर अट्रॅक्टिव्ह.
आपण चेंज करू शकतो ते टेम्परेचर.
हाँ,
तर बघा, नैसर्गिक गुळ म्हणून
color
marketing
is
करता
also
येईल ना.
due to the
यांचे चार
chemicals
वेगळे पांढरा
present.
पण गुळ दिसतो यांचा.
केमिकल्स ऍड करतात त्याच्यावरती डिपेंड आहे ना. बेसिकली तुम्ही विदाउट केमिकल जर जॅगरी काढला तर तो डार्क चॉकलेट कलर मध्ये जाईल.
तीच बघायची आम्हाला सवय.
तेच पण फक्त बघितलीच पाहिजे.
आता-
राबू खाला तर त्याला थोडं साखरेची टेस्ट.
Sir, I think basically you have raised 2 points. Number one is the time limit. I mean, there is no question that it will require less time, but most important thing, labor availability, because it is totally dependent on the labor. So you can say strongly that it will save the dependency on the labor. Number one. Number two, depend on the flow, different shapes can be made, but only thing is that what about the nutritional comparison? Say for example 267 degrees centigrade temperature may hamper some of the micronutrients. Okay, so that you will have to check or compare with the traditional way of made jaggery and by this method. So I think these two can be compared.
तो 40 रुपये किलो आपल्याला जर बाहेरचा मिळतो तर मायक्रो रिॲक्टर मध्ये केलेला 25 रुपये की 60 रुपये की 200 रुपये पर किलो.
सर सध्या त्याचेच कॅल्क्युलेशन आणि त्याच्यावरच आम्ही काम करतोय.
बट व्हॉट इज युअर गेस्टिमेशन?
230 सर सध्या 250 kgला 2000ची इलेक्ट्रिसिटी लागते.
नाही बट ऑर्डर ऑफ मॅग्निट्यूड 200 रुपये और 1000 रुपये किलो. आय मीन यू कॅन टेल दॅट ना.
नैसर्गिक गुळाच्या जवळ जाणारी किंमत हवी. दॅट्स व्हॉट ही इज हिंटिंग ॲट बेसिकली. तर लोक ऍक्सेप्ट करतील.
It means 8 rupees per kg of jaggery.
Yes.
Will be the cost of electricity.
इलेक्ट्रिसिटी.
बरोबर ना?
हां.
म्हणजे 2000 बघितलं तर 250 केलं तर ऑलमोस्ट 8 रुपीज येतात.
Electric efficiency चांगली असू शकेल.
Maybe. बरोबर.
कारण
Last
heat transfer areas is very high compared to conventional.
Correct.
And शेवटी all that heat required is to remove water.
Correct.
Is the latent heat of evaporation is what you have to, so the more efficiently you remove the water, the cheaper it is going to be. So that is where the USP lies in the, as far as the technology is concerned. I can see the point. Ultimately, that capex opex calculation will have to be done. And understand what is the real problem and I'm getting the feel that labor is the main issue. It very much might be, but is that the only selling point? And are there other values that you can add? Nutritional is one, color is another, different shapes of jaggery could be the third. There could be many things you could make the technology. You have to find the pulse of it.
They are in the right direction.
Right.
Yeah.
So we'll have one more presentation on jaggery from Professor Mahazani of IIT Bombay, and then after that, Professor Saraf, maybe we can.
Yeah, thank you. Now we move to Dr. Sharda Devre from Government College of Pharmacy in Amravati, talking about neuroprotective herbal oral strips for Alzheimer's disease.
Good afternoon to respected members of RJSTC, MCCI, Lele sir, all the academic and industry dignitaries. My project is related to the neuroprotective herbal oral strips, especially for the Alzheimer's disease. Why Alzheimer's disease has been chosen? Because it is the thrust area in most of the funding agencies also, and even the government is also highlighting these particular areas. Another thing is that there is a 1 to 9% burden throughout the world in different parts of the, in different countries of the world. There is an economic burden also, about $2 trillion, and that will be up to the 2030 only, means just within these 8 years. And all we know that the Alzheimer's causes the dementia, and social life, the quality of life of patient as well as family get affected. And it has the lot of health care burden also. There are a lot of things which are involved when there is a neurodegeneration, and especially for the Alzheimer's disease. And based on that, we have selected our traditional knowledge, that is from the Ayurveda, Siddha and Unani system, the two very popular drugs, that is Brahmi and Jal Brahmi, that is Bacopa monnieri and Centella asiatica, which are found to be, means they are Medhya Rasayana, they are acting on the brain, they are responsible for the memory enhancement. Obviously, in none of the medicine system, traditional medicine system, Alzheimer's disease is not mentioned. So it becomes very necessary to evaluate these particular selected drugs. There are a lot of Medhya Rasayanas, but these two are more prominent, and these two drugs are chosen. They are chosen, means they are studied for the different preclinical studies, toxicity studies. Further, why not the tablets and capsules or other dosage forms we have selected? Instead that we have selected these oral films, which are the thin films. Because Alzheimer's patients, that actually starts from the age of 55 to, as life expectancy is increased, so it is up to the 80 years old. So irritation is the most important thing that has been observed in the age-old peoples, and tablet capsules, as well as the size of these due to the high doses of the various Ayurvedic drugs, that leads to the issue, means it is not the patient friendly. So to improve that also, this oral film dosage form is selected. Then there are lot of chances to improvise it by various modifications once it is get under the manufacturing level. These two plants chosen, they are traditional extract as well as the marketed extract. By CDRI laboratory also there is one extract, CDRI 08 for the Bacopa monnieri, which is in a market and utilized in by the various companies, and have the claim of Alzheimer's disease also. Our claim is related to the neuroprotection as well as Alzheimer's disease. Various extracts, at least 15 to 20 different extracts and different solvent combinations were utilized and compared with the marketed extracts also to find out the bioactive fractions as well as the its efficacy. Similarly, it is done for the another important drug, that is a Centella asiatica, and again the bioactive rich fractions compared with the Subsina, that is Sami Labs product, that is Santalene, and we have tried how to improve the content of the bioactives so that dose reduction should be there and fast relief, or there must be the relief to the patients. All these 3-4 patents we have filed related to the extraction procedures of the Santal as well as the Bacopa. Here with some study that we have did for the preclinical acute toxicity in vivo and in vitro Alzheimer's disease, especially for the neuroinflammation, which is the responsible reason. Then another, it is the acetylcholinesterase, means neurotransmitter secretions, and further related to the antioxidant and other preclinical studies. Then we have developed the oral films of loading and optimization of the film formulations as per the suggestions during the review meeting from the RGSTC by respected Sapre sir and Kakodkar sir and all members, that the polymer should be natural instead of synthetic. So according to that also we have conducted the trials and we have developed this formulae for the addition of the extracts. Both there are 4 types of the extracts are available, 2-3 extracts as per the sir. We have given the name scent memory and Baco memory to the extracts as well as to the oral films. As per the respected Prashant sir said that readiness and manufacturing level, and Lele sir, respected Lele sir, that is TRL level. So according to that we have tried to be in contact, continuously in contact from the start of the submission of the project even up to this. We are in contact with the 2 important companies that are manufacturing the oral films. One is Unijewels from the Nagpur and another is the Avishkar oral films, that is from the Hyderabad. And according to that we are planning, we have tried its trials, and as per the suggestions from the RGCC members, especially Sapre sir and Kakodkar sir, that social needs and social, that is also said by Soni sir, that local resources and business generation should be there. So Konkan, Igatpuri or other Gadchiroli regions, farmers can take up the cultivation of the both Brahmi and can avail the raw material. It can be avail from the different parts of the India also, and accordingly raw materials, then extract, we have the 2 technology. First, that is the extracts, that standardized extracts which have the very better activity for the Alzheimer's related complications, and according to that we need such a extract companies that will able to manufacture the extracts that we have standardized. And next it is the oral film manufacturing. Third, it will be the marketing. So 3-4 companies type, or if RGSTC permits, and we are in contact with Tikekar sir, and as per suggestions of that, academic startup can be initiated, and as per that said by respected Shah sir, that Industry Institute interactions, we are from the start of this, for this project we are in a start. For getting the results, as it is a claim, it is nutraceutical, it is not directly for the treatment. So according to that 4 to 6 weeks initially, and then further again 4 to 6 weeks by the experts can be, they can, doctors can suggest it. Yes, there are certain side effects to this extracts, that are the stomach upset is observed, diarrhea like conditions, but that are not the common, it is not routinely observed in the preclinical studies, and that is already well known also. So accordingly also we can come upon that. We are in contact with, as we are from pharmacy and especially from government, so we are in contact with the alumni that are working in the FDA. So based on that, what are the licensing procedure, FSSAI licensing or the Ayush ministry related that licensing, we are in plan that, and for further, as per the guidance of the RGSTC and Tikekar sir, we will move towards that, and today also with the guidance.
Thank you so much.
Yes sir, it is preventive also and curative also sir, both.
Okay.
So that's why we can, in a nutraceutical doses, we can start it for the prevention if it is early detected. And one important thing, sir, that this particular plant extract cannot be only restricted to the Alzheimer's. It can be related to the all neurodegenerative disease also. So accordingly also we can plan that for. It is suggested by the companies itself.
And your proposed formulation, how does it compare vis-a-vis the existing?
No formulation, means whatever the formulations available, churna or like that traditional dosage forms or tablet dosage forms that are available. Some of the medicinal plants oral films are available, might be in airports, or that might be, you have seen the certain boxes, even gifting boxes to the children with the vitamin and all that sources that are available in the market. Ashwagandha oral films are available. Brahmi oral films that are planned by the Unijules itself, but I don't know what's the reason they have not, means that is not in a market, sir, now. And we are in a contact with the Avishkar oral films, so they are going to plan it, means launch it from the point of view of Alzheimer's disease first, and then for the other neurodegeneration.
So last question is, did you get to do any clinical trials?
Yes, sir. When means there are the two combinations for which I will not need, because it will just need the FSSI license, and based on the traditional claims and the data of the preclinical, that is toxicity studies, we can start its marketing. But for two extracts, I will need the clinical trials, and that also I have discussed in the last review meeting with the RGSTC, and even I have mentioned in the letters communication to the RGSTC that we can initiate the clinical trials also. And for
the end user, you know,
yes.
you may get a license
Yes.
from FDA,
Yes.
but for an end user, let's say I am prone to get Alzheimer's, and I want to know its merit,
Yes.
so to
Yes.
put it on your label, for example, that
Yes, sir.
it works or
Yes, sir.
something.
As per guidelines of the clinical trials and Schedule Y, means what we need, if it is strongly claimed, if it is traditional drugs, and we are using in that same type of the composition without altering too much, then based on the available data, literature data, available clinical, from clinicaltrial.gov.in, from that data and from the preclinical studies, we can start marketing. But if there is anything changed, which is changed in the standardized extract, which are comparable to the CDRI 08 and the Centellin of the Subsina, I need clinical trials.
So you will be able to sort of put that claim on your
Yes,
packet,
yes,
thing.
yes. And for that, before initiating the clinical trials, I need the GMP certified manufactured means oral films, which sir rightly said that why our institutes, means institutes are not with that facility. And when I have to start the, means doctors are ready, we are in contact with them, but we need the GMP certified products, so that they can give it to the patients, and all that procedure also to file the clinical trial initiation forms and all these things.
Ma'am, I have 2-3 observations. Am I audible to you?
Yes.
Yeah, this is regarding if you are going to conduct a clinical trial, I would like to ask if it is compared with the existing only Ayurvedic preparations or allopathy medicines which are already there for Alzheimer are being compared with the with your formulation.
Yes, ma'am. There means randomized trials, and in that we can add it, and we must add it, ma'am. Only one galantamine is there, and other 2-3 synthetic drugs are there, and that are not targeting to the Alzheimer's directly, that are targeting to the symptoms, like that acetylcholinesterase secretions and all this.
So the existing formulation or your formulation is at par with the existing allopathy as well as Ayurvedic preparation, Ayurvedic medicine.
Yes, madam. But that's why it is necessary to start or initiate the clinical trials to complete it. Why?
True.
Because we will get the idea. Means
Yeah.
in nutraceutical level it is going to be best. But from the comparison with the synthetic drugs, we have to do the clinical trials.
Yeah, and if you are trying with the Unijules at Nagpur, I can help you to initiate that contact, or I can help you to take it further in terms of your manufacturing. That I can do. And one more thing, you mentioned about your extract manufacturing, and you need GMP certified manufacturing unit where your formulation can be a GMP certified formulation.
Yes.
Even for that purpose, I can give you 2-3 contacts which can take further your manufacturing for extracting.
Thank you so much, ma'am.
I think I
This
can
is-
add one small thing.
Yes.
So when we compare with the risperidone or any other medicine which is
Yeah,
available,
yeah.
allopathic, I think the Ayurvedic would always, we can put a label claim as not therapeutic, but probably in the disease maintenance. Because I think therapeutic would be a very ambitious claim.
Yeah, by
It could
FDA it is not even allowed.
Yes, we
For
cannot put
FDA
that.
it
So
is
it
not
will be
allowed
disease
to put
management.
therapeutic. Yeah,
Correct.
you can mention as it can maintain the disease or helpful in so and so disease. That's it.
Supplementary.
Yeah,
As a supplement.
As a supplement, absolutely.
Yes.
You mentioned
two technologies that are going to claim, that is nutraceutical for preventive, like that, and 2 technologies which are rich in bioactives and having the very different extraction as well as the procedure than that of the traditional methods will need the clinical trials and then therapeutic claim.
Yeah.
Madam, can I just add one more point? Basically, if you are also thinking of prophylactic, it will be a good idea to factor that into your clinical trial, because clinical trial
Yes.
designed for prophylaxis and therapeutic would be totally different.
If the drug is not well known, then Ayush Ministry means we have got the suggestions from that. It is well known, thousands of years of evidences are there, so you can go for that. Dosage form related GMP certification is only we required to certify it, to allow it to in a market.
But I would suggest to go for at least a small group of people,
Yes.
50 patients.
Yes,
So
that.
I have 2 questions.
Yes, yes, sir.
One is,
Can we take it offline, please? We are running a little behind schedule now.
Thank you so much, sir. Thank you.
Let me request Dr. Munira Momin from Nanavati College of Pharmacy. She will make a presentation about nano mule gel for arthritic inflammation of the ethno-medicinal plants of Western India. Over to you.
I will be presenting both, representing Dr. Padmini as well as myself. Dr. Padmini, because of some personal pre-commitment, she could not join today.
So she will be presenting on the forms and hydrogel sponges for
Yes.
burns and diabetic wounds
Yeah.
as well.
So as per the sequence, I'll go for first with the hydrogel
Padmini.
of Dr. Padmini. So Dr. Padmini is a principal investigator. So continuing with taking the thread from the earlier presenter, where she has also used Centella asiatica. She has used Centellin for the neuroprotective. Asiaticoside is another chemical which is very well kind of application from Ayurvedic point of view. In ancient time, they were using only water extract. It's a water soluble water extract they were using, putting on the wound, because it improves granulation of wound healing process, which is extremely important for preparing the ECM, extracellular matrix, and then closing the wound. So that's the reason we took the asiaticoside from acetic acid and curcumin, which is very well known from time immemorial. We have been all using curcumin in a different forms. Whenever there is a cut, we just put curcumin. So we have used a pure curcumin. So this.
Can you make it on full screen, please?
Oh, sorry. I think I'm sorry. Yeah. So what we did is we designed the entire protocol based on pharma industry, keeping pharma industry in our mind. So we conducted basic pre-formulation study, because that's very important for a long-term stability and selection of the excipient. After that, we also did a formulation evaluation for validation of the drug content. It's extremely important for reproducibility, establishing the reproducibility as well as efficacy of the formulation. So we developed HPLC method, which is very sensitive, and it is widely accepted, US FDA as well as the Indian pharma. HPLC is the one which we use. So we developed simultaneous estimation method of acetic acid and curcumin. Acetic acid was estimated as per asiaticoside. Solubility was required because curcumin is a poorly soluble, as we all know. So we had to use beta-cyclodextrin, which is very well used as a solubility enhancer in pharma industry. We did a dose selection. because we wanted to optimize the cost also, and that's why we wanted to see which is the lowest dose which would be used for the product for getting the maximum output. So I will share that scratch test which we have done, in vitro scratch test. We developed two products. One is a hydrogel and another one is a thermosensitive gel. Now the reason of you developing gel, thermosensitive gel, is especially for burn. So when there are burn patients, the skin becomes very sensitive, and when we put any gauze or any dressing, even hydrogel, at the time of removing, it's very traumatic because the skin is still into that healing stage. So what we developed is a spray. So it's a foam spray. When we spray, it comes out as like that shaving foam. So it is a foam, and then it slowly settles on the wound, and then it covers the wound, and then it automatically gets biodegraded, and we don't have to remove. And that's how we can reduce the trauma. And then we did a lot of evaluation for final formulation evaluation, and then we also conducted in vivo animal model for burn as well as wound model. We could not conduct diabetic model, foot ulcer model. So there were 6 antioxidant activity studies we conducted, which are very well known. Out of 6, 4 studies give a suggestion that it is a synergistic effect. So you can see this was the product. This is a hydrogel. The word hydrogel is because it has the ability to absorb lot of moisture, the exudate which comes out of the wound, and that's how it keeps the wound bed dry. And it has a capacity around 100% weight of its own. It can absorb the exudate. So this was the HPMC, which is a good film former. Sodium alginate. Alginate is also helping in wound healing process. Natural polymer, and we have used soy lecithin to stabilize the entire formulation into hydrogel. We used a design of experiment statistical model. This is the spray, where also we used a Methocel 5M, low viscosity Methocel, and Poloxamer is at a body temperature it converts into gel. So when it is in a bottle, it is liquid, and when we convert into spray, when it sets on the wound at a body temperature 37, it converts into gel, and that's how it gives a nice spongy layer on the wound. We conducted scratch assay, as I said, in order to do the doses. So you can see the 5 microgram and 7.5 microgram combination of C plus A gave a very good, like 99% after 24 hours assay. It's basically monolayer cells. We scratch them in the center, and then we allow them to heal in presence of different concentration, and we see which one kind of gets back to normal and heal, and that's called a scratch test. So L929 monolayer cell lines were used. This is the study, and we also conducted drug release, because it is very essential to see that drug release is also extended. We don't want drug to be coming out immediately. The hydrogel was also studied for its disintegration pattern. It should not disintegrate or dissolve into the wound bed very fast. So it was a 24-hour, easily we could see that the wound bed, the gel was there, and then slowly releasing the drug. And this is the burn model which we conducted. You can see that it was compared with a different concentration. So we had a drug-loaded sponge, and the last one is a drug-loaded thermosensitive gel. And you can see 98 to 94% day 21 drug, the wound contraction was there. I'll show you more pictorial. That would be very easy to understand. So you can see group number 6, which is a hydrogel, and you can see group number 8, which is spray. So the spray has a faster healing. If you see day 15 and day 21, compare the 6th and the 7th. The 6th one still has a open wound, while the spray has the wound has been closed. Same way the wound model, and this was the burn model, and this was the wound model in both ways. In wound model, which was a cut, the skin was cut, the hydrogel, and both of them they showed a good closure. This picture is a histopath. You can see that last one in the corner, there are a lot of fibroblasts have started coming up. That is a good indication that the wound is getting healed properly. The technology transfer was done in the Mill laboratory at Baroda. We prepared the placebo without drug hydrogel and with drug, both this, and we could successfully prepare the big size hydrogel sheets. The Mill laboratory is very much into hemostats, gelatin and all. They are mainly, their market products are gauze, primary gauzes, secondary gauzes, but without drug. So they were like more interested in looking at our hydrogel sheet. So we prepared hydrogel sheet as well as the drug loaded hydrogel. In completely 2 days, the student was there and we worked on that. We did presentation. The student had got the Avishkar award. The patent has also been filed. The hearing is completed. Hopefully very soon we should be getting the result of patent. It is, the hearing is completed. It's almost 4 months the hearing is completed. So with this, we sincerely thank RGSTC for funding this project. So technology, so in terms of technology transfer check, yes, we have done the technology transfer scale up in the laboratory, in the industry setup. We have not yet identified any potential industry collaborator. Thank you.
So ma'am, there are multiple burn wound products available, right? So how do we compare yours with what is already available in the market? There are so many of them that people get confused which one is the right one to use. And
Right. So that's the right question. We actually met, I forgot the name, right now it just went off my mind, for Airoli Burn Center, which is the largest burn center. I forgot the name of the doctor, very famous, Keswani, Dr. Keswani. So we met Dr. Keswani and he said, right now, he said the product is good because the hydrogel is what they are looking at. Even they are using hydrogels without any drug, basically to keep the wound dry. So they say the most important is what we need is a sponge which is there, the thicker sponge which can absorb more exudate. They don't have to remove. The moment we said that we have a thermostatic thermogel foam, they say it would be good, but he has not yet kind of committed that he would help us. Yes, there are a lot of products available in the market. We don't say that we have yet not compared the first point of any product which is available in the market for burn. We were looking at the technology possibly which could help burn patient, that is our foam gel. So we have not compared it yet. So hydrogel would help, that is our kind of a hypothesis, because it can absorb exudate, and a foam would help because it gives a nice spongy bed which we don't have to remove them. It just gets kind of lysozymes which comes out of exudate would break it.
are the actives the unique one for the acetic acid and
yes, the
curcumin? Curcumin is of course
but
known.
acetic acid, the reason we selected is because it helps in granulation. We found one patent and not more. One of the DST project based patent we found. So it helps in granulation process. So it hastens the granulation. That was the main rationale of using it.
For the uninitiated ones like me, often at times they say if you have a burn, just take your toothpaste and put it.
Should not. Should not.
No good?
No. It is just because you're putting toothpaste, because it has a menthol and all, it gives cooling effect. Should not be doing it. Recently we had a first aid workshop, burn workshop specially, and we had invited one doctor who was in the Navy group for burn, and he said never ever use toothpaste on your burn.
How about banana skins?
No.
No.
Madam,
Banana skin would help, the reason I'm telling you.
Madam,
it has a polysaccharide.
Madam, I have a question. So first is, when you talk of burns, what is the degree of burns that this can address? And second is, even with the previous speaker, when you conduct clinical studies, what is the quantum of customers or potential test samples that you study before claiming that it heals faster?
So generally what we go, and this is what I have done in my current, the second one which I am going to present. So generally when we have a good animal data, it is for first degree. What we studied for animal is only first degree burn. We have not, because that's what Dr. Keswani said, that do not claim any of your product for secondary, tertiary burn, because that altogether requires a different treatment. So the first degree burn we used in animal study, we could see the good result. And as per Dr. Keswani's discussion, he said probably you can start with only 2 patients, because burn is something which you do not want to play with.
Correct. So essentially, I think there will be some human studies required before you can genuinely market it.
Yes.
And you will need certain quantum of data before you can make claims that.
Yeah, very much. We cannot claim right now that we can directly go for market. We have to conduct critically clinical trials.
And then the challenge is, when I get burnt, let's say in my kitchen, I don't know whether it's a first degree or a second degree. I want the best medicine at that juncture. Most likely it is beyond the first degree, most burns, I would imagine. First degree burns are probably when I touch something hot for maybe
And then
5
inside the
seconds.
blister.
Yeah.
Yeah.
So, I mean, you will need I think Dr. Lele pointed out, you will need a comparison with
Yes.
what's available in the
Yeah,
market.
we definitely did. So we have not done any market comparison, as well as we have not yet thought of any of human clinical trials. Yeah, we would not go for a secondary trial in the beginning. A secondary burn, we would only go for the primary burn first, and that would be under the strict observation of expert. Because in burn, there is a skin and a protein which gets kind of, there is a
Yeah.
deep wound.
Yeah, but the challenge will be to find someone with only a first degree burn, right?
No, we can. I mean, first degree there are lot, because mechanic into the garage and all, they get first, second degree initial burn when they are dealing with the machines, hot and all that. They will get.
We will take it offline.
Yes.
Next one, please.
All right. Thank you very much. So I'll just quickly go on the second project, and we have up to certain extent established the efficacy of nanoemulsion. So this is the nanoemulgel which I have been using for my frozen shoulder and the pain. So and it works. So efficacy study of nanoemulgel. The basic idea was the genesis of this project is when we started talking with Dr. Ashok Vaidya, who is a very well-known pharmacologist, known as a reverse pharmacologist, father of reverse pharmacology, who has worked on a lot of Ayurvedic molecule from bed to bench. So reverse. So whatever the established Ayurvedic products are there, well proven in a hospital setup, get back to lab, standardize it, analyze it, reproduce it, and then generate the data, and then in a more scientific way we can produce from pharmaceutical point of view. So that was the whole idea. We know in our family, all around in society, around 20%, 30% people after 55, 56 would be somewhere either having osteoarthritis or arthritis or a joint pain or joint inflammation, and that's when we use generally the Volini gel, which is a diclofenac, non-steroidal anti-inflammatory, or we use lot of other Ayurvedic product, the oils, which are not that comfortable to use when it is a oil, like it stains or something, it spoils the clothes. So what we used is we have prepared the emul gel. So emulsion converted into nano droplets, put it into the gel. So the nano droplet will help in a further penetration. This is which is most important. It has to cross through the skin barrier, stays either into the synovial fluid, and then releases the drug, and that's how reduces the inflammatory markers. So we worked on the pro-inflammatory marker as well as the post-inflammatory markers also. We know nanoformulation has, nanotechnology based product has picked up from 1990. Oh, sorry. I'm extremely sorry. Yeah, sorry. Yeah. So we have US FDA has given approval since 1990 to now. If you see, there is an exponential increase into nanotechnology based product. We as an Indian and Asian, we have a very rich heritage of natural compound which have been used as a medicine, can be converted into nano. Lot of them have been used for a lot of different ailments. Capsaicin even has used very much into rheumatoid arthritis. The only drawback of capsaicin products are available. Only drawback is it's very irritant if you have not washed your hands properly, the eye and lips and all, the sensitive part of the body would get tremendous in pain, and that itch would be there. So what we have selected is the ginger, which is ginger oleoresin, which is very well used. We have combined with the gugulosterol. So the combination of these has been picked up from the Nimitli report, which says that the Western Ghat folk healers, they used lot of Commiphora gugul. So Shalaki, which is available in the market, is a different gugul. The Commiphora gugul has been used by folk healers in a Western Ghat. Ginger is used, ginger oleoresin and ginger oil has a very good anti-inflammatory activity. what we used is castor oil as a oil, which is a functional oil. So if we look at gingerol, it works as a 5-lipoxygenase inhibitor as well as a COX-1 and COX-2 inhibitor, which is a glucosterone. We use a functional oil. Castor oil, I think some of our elders would say just apply castor oil. And then we apply in the joints. It works because of the racinoic acid, but castor oil, because of thickness, it doesn't penetrate well through the skin, and that's where we converted into tiny droplets. We were at some point thinking of fenugreek oil, methi oil. We take methi orally, but the methi oil also works, but we dropped that idea because there are too many actives were coming, and that's where we only use a racinoic acid and a glucosterol and a ginger extract. These were the batches which we prepared. I want you to pay attention on the Jita size. You can see that so we used oil mix, one synthetic oil and one which is all pharmaceutical grade allowed, and we use castor oil with some surfactant which is going to give a stability to my nano oil droplets, which is of 18 nanometer size, and they were kind of a very well polydispersed, and we can see the stability is zeta potential. We could see the drug content was very well. This is the product which, so the product I can change the consistency because I wanted it for myself, for my shoulder. It was much easier if it is consistency is less. So I converted into a little less consistency, but it can be converted into thick consistency as a gel just for the fingers and all. You can see the droplets, that is a TAM image of nano emulsion. We conducted the drug permeability diffusion through the skin. So we used the ear skin as well as semi-permeable membrane, and we could see the sustained effect of the drug release. We also conducted sensitivity because we know the capsaicin and all are very, very irritant to the skin. We wanted to see our product, so we used the HETChem study on the fertile egg, and we could see our product was no irritation at all. We also conducted on a raw cell the anti-inflammatory study. Paw edema measurement was conducted. X-ray analysis we did for the paw edema of rat, and you can see the product which was applied, that the inflammation reduced. We did histopathology, and we could see a lot of infiltration of inflammatory cells were reduced. This is again L10 blood plasma concentration, because ultimately what we need is the synovial fluid as well as blood. So both the concentration we had seen, that inflammatory markers has to come down. So one was in the blood plasma and another is a synovial fluid, because that's where the joint pain synovial fluid becomes very thick because of lot of inflammatory markers, and we could see that the synovial fluid viscosity reduced as well as the inflammatory markers reduced. So you can see that pro-inflammatory marker as well as the inflammatory marker IL-10 were also reduced. So we can see that anti-inflammatory markers, cytokines, IL-10 reduced, and which was compared with the diclofenac, which is the NSAIDs, non-steroidal anti-inflammatory, which is very widely used in form of Volini nanoemul gel. So that is also nanoemul gel is available for the diclofenac, that is a Volini gel. Stability study, this product is almost 6 months old, and I've been using since 4 months, so I use this much, half of the bottle, and you don't need to use much. We join hands with the doctor who is a specialist in the rheumatoid arthritis, Dr. Ashwini Raut, and we gave them the protocol that is a 1 toothbrush size gel, has to be applied twice on knee. If it is on finger, it has to be half of the toothbrush, and slowly rub, entire gel absorbed, and it gets absorbed because it's an aqueous carbopol gel. And 15 days we wanted to see the results. I'm going to show you the table. Expected observation were reduction into the inflammation and pain. So these are the data of our first set. So we prepared from lab scale of 200 gram to 2 kg gel, and we gave it to 10 patients, 2 tubes, and we asked them to give their reports. So every 3rd day and 15th day they gave us the report. You can see the different age group of 27 to 65. I have not included mine. Even doctor himself used. So for a different inflammatory condition he has used, for shoulder, for knee, for thumb, for wrist, for rheumatoid arthritis, for only capsulitis, that is generally for the frozen shoulder. He has tried for all different conditions, and we could see that. So E is excellent, G is good, S is satisfactory. So we can see the lot of patients, they have given Even tendonitis gave a good result because that's an inflammation of the wrist. 3 days, 15 days, and some of the patients were not on anything, some of the patients were already on something, which is NSAIDs or other. So the future plan is myself and Dr. Tabassum, we have joined hands with one of the students. We have registered the company, Global Health Solutions, and we are going to go to IIT. Thanks to RGSTC for providing this opportunity. He is our student as alumni, so we are going to work on this project. One is a nutraceutical. We are thinking of third-party manufacturing. The technology transfer is possible. We did at a lab scale to 2. What we need in industry is only a mixture. The stainless steel mixture, nothing else, one pot product. You just mix oil, surfactant and ingredients, mix it, put a gelling product. So how it goes in pharma industry is you have an overnight soaking of carbopol into water and you have a gel, and then you put all this oil mixture into that and the product into the actives into that. Slowly anchor mixing would take place because and then convert into gel with a change in the pH. This anchor mixture will give a nice mixing without any air bubbles, and that's it, it's ready for packaging. The cost approximately, we counted around 20 rupees. The currently market product which we see is somewhere around kind of a 80 rupees to 150. I'm going on the very lower side, the regular products, rather than the sprays and lot of other products which are available for the joint pains. So with this, and it has been patented, hearing would be soon. We have submitted all the queries which they had given. We had also submitted the National Biodiversity Certification also. And thanks. Question, yes.
Dr. Mundira, did you by chance try your hands with Vitex Nirgundas, that is that Nirgundi?
No.
Which is traditionally tribals had been using, and there are some oils in the market which use the extract of that. So maybe you want to sort of put your nano.
We can do that. So it's a platform technology. We can put oil, we can put other oil extract as well as water extract. So because it's an emulsion, it has an oil as well as aqueous phase. So it's a platform technology, we can mix anything.
We can help you get the extract.
Sure, and we can join hands with Ashwini Raut. He is very much interested in this kind of product because he himself used one. The last patient was him. And then he said that give me more. In fact, my orthopedic MD ortho, he said that send me the bottle because you are saying that you don't need now further MRI. Yes.
Ma'am, don't we need clinical trial permission for all this which you have done?
So same thing, it is already listed into Ayurvedic listed drugs, safe drugs. So what we are doing is a small pool. So we started with 10 patients, now we are going on 15 patients, and after that we are going to go on 50 patients.
Madam, how do you measure the efficacy? This is not a therapeutic cure. This is probably some form of relief from the pain. So do you measure efficacy in terms of relief for these many hours or days, or how do you measure the efficacy and compare it with the existing solutions or other alternatives?
Yeah, sure. So what we measured in animal is with diclofenac. So first group we gave 10 patients whatever existing situation and medicines they are on. We wanted that is it add-on helping, number one. The second group whom Dr. Ashwini has selected is those who are not on anything. So he is going to give it to only those patients who are on NSAIDs, diclofenac and other celecoxibs. He has asked them to be off for 15 days and use only this in order to see the efficacy. Now the third group which we will go, we will have 2 groups, one on celecoxib and another on this, and we will see their patients' clinical exploration only.
But what is the unit? Is it time of relief, or how do you measure it? So let's say I have arthritic pain, for example. I use a certain kind of a certain kind of oil, and I feel relief for, let's say, 48 hours, and then I have to reapply the oil. So is that how you measure the efficacy in this case?
So what patients have told us in this, so after 3 days, they just gave their, their observation that yes, I have a pain relief. So this is right now the basic status of clinical. It just says yes, there is a relief. Yes, there is a relief in inflammation, which is visibly Dr. Ashwini could see that there is a relief. There is no, what we should be doing when we go for actual clinical trial, we would be checking the patient's blood profile, we would be checking the inflammatory markers in blood. That would be the real clinical trial with the two groups, one on this and one on COX inhibitors.
And then you mentioned this can be just mixed together and stirred, etc. So are you targeting a B2B or a B2C? Because if you give me the raw materials, I can mix it at my home and use it.
It depends. So it could be, so if we go for a production, it will be B2C, right? It's like that.
Okay, so that has not been decided yet.
Yeah, and to be very frank, if you ask me about entrepreneurial side, you will have to train me in IIT to understand that.
No, and third question I have is, there are newer stem cell therapies coming up for arthritis and other things. Are these compatible or maybe better than those?
Compatible.
Compatible.
We can further improve, like what rightly sir said. So what we say, whatever we don't want to make it a polyherbal. It's difficult to analyze using HPLC, right? So if I add one more Nirgundi, I will first see that can I develop analytical method, simultaneous estimation of these three main ingredients which are measurable, which I can measure in a blood plasma, and I can measure my IL-6, IL-10.
Is ginger oil is used or ginger oil is used?
Ginger oleoresin.
Ginger?
Not oil. Oleoresin. It's a oleoresin. It's a oleoresin which has a effect.
Then ginger oil, shagol as well as gingiberine, and there are more than 100 compounds.
But gingerol, which is more into oleoresin, ginger oil is not that effective. This is the input which I got from Dr. Ashok Vaidya.
So one question.
Dr. Munira, Dr. Sapre will tell you more, but RGSTC is planning to take a larger, big umbrella project. I think Dr. Ashwini Raut is a member of that.
Yes.
And so maybe in 2 words you could tell.
No, but that is a new project which we are planning for a
Plant
kind of integrated, data generation on selected medicinal plants, where you go right from the level of, say, cultivation, harvesting, processing, plant chemistry, analysis, and even the further processing, chemical engineering part of this, up to a commercialized. So this will be some kind of a multi-institutional program, which we are thinking about, and we are.
Sure. We'll be happy to be a part of it.
Yeah, yeah.
Sure.
So we may be involving some of you.
Sure.
Thank you.
Sure.
Thank you.
Thank you, sir.
Let us now move to Dr. Sanjay Mahajani from IIT Bombay. Again, he is presenting the R&D in Jaggery Process and Product Development project.
So thank you very much, sir.
Thank you.
So good, I don't have to give much background because already Professor Saraf has already talked about what Jaggery is, and so we have a project sponsored by RGSTC, and it's going on for so many years now, and we have been looking at various aspects of Jaggery making, right from process to product. So I'll just tell you a few of the observations or innovations that we have made, and if you have other questions, we can probably discuss offline. So if you look at a Jaggery making process, the crushing, then you have evaporation. This is a traditional process, by the way. Then you have clarification, then evaporation, and then once the juice is concentrated, it is crystallized, and then you have molding, and then finally you take it to the market. Now it's a highly labor intensive process, and it is not just the evaporation that is labor intensive, but other processes, other steps in making Jaggery, they are also labor intensive, as you can see from this. So some 7-8 years back, like our team did a survey in the field and tried to identify the problems that Jaggery makers are facing, and we rated them based on the seriousness or the intensity, and Jaggery making labor requirement in Jaggery making was one of the top ones. And of course, the kind of product that consumer is using today is also not of good quality, and that's the reason Jaggery market is going down day by day. and sugar is kind of replacing it. Of course, recent trends are slightly different because of health awareness. So what we thought that we'll try and make technological interventions in almost each of these steps and try and automate jaggery making process. Of course, it will be capital intensive, but then of course that can be compensated by taking the scale to a larger scale, and we can have economy of scale advantage there. So that is one thing. Second possibility is, so on the left hand side you can see, on my left you can see a jaggery making plant, a bigger unit, a large scale unit. In the middle you can see the conventional unit where we want to make some interventions as well. So we are not saying that the conventional unit should be replaced by the big units or something, because everything has its own merit, okay, and we have to, in the beginning stage, like we'll have to really explore each one of them. And on the right hand side we have come with an innovative way of making jaggery, like it's in the mobile plant. So we have tried to kind of put all the steps or all the machinery on a mobile unit, okay, so that we can take advantage of the fact that a unit can go from farm to farm and make jaggery. And instead of transporting cane, you can transport jaggery, which is 1/10 of cane in terms of volumes. I'll just move fast. I'll tell you what modifications we have done. So a normal clarification process, it's quite tedious. Like we'll have to add many additives and remove proteins, phospholipids, and many other impurities from cane, which impart color and bad taste to jaggery. So that is to be removed, and we came up with a continuous process for removing these impurities. Of course, we took hints from the sugar clarification process, and we were able to kind of implement it. Then the second one is the heating system. Of course, on the left hand side you can see the way heating is done in the conventional units. We replaced that with the help of thermic oil. Thermic oil is heated at another place, like a boiler rather, wherein we are burning bagasse. Then the crystallization process, this is very important. The upper one is the normal way of doing it. Now we came up with a screw conveyor which works also as a crystallizer, and this is a very important step in the sense that the final texture of the product would depend on how you do it. So just evaporating clarified sugarcane juice is not enough. We need to really work on this particular step, and we took long time to really understand what happens here. Okay, so it's simultaneous solidification, evaporation, and crystallization, the three steps happening together. So scaling up is not so easy. We have to have right parameters there. Then molding, this is the conventional way of doing it. We tried to automate it. Of course, it took some time to do all these calculations and get the things in the right place. Bagasse drying, this is a conventional way of doing it. Large amount of land required here, and then the labor also is quite labor intensive process. We put the bagasse dryer. Again, nothing. It's not rocket science. But then we had to kind of design it for the conditions that we are working in. So the flue gas that comes from the burner is used to dry the bagasse, which goes to the boiler, and we save a lot of land because of this, and labor also is going down. Then further to increase energy saving or reduce the energy requirement, we again took hint from sugar factories and tried to install multiple effective evaporator, and successfully it is functional. In fact, it's going on at present. Sugar season has just started. So energy consumption has gone down. Then what's important here is like the conventional products that people use in the form of blocks and all, they are not quite conducive in terms of the usage in kitchen and their shelf life and so on. So that's the reason. That's another reason why jaggery is becoming unpopular. So one way is to kind of get it in the right form, and powder is becoming quite popular today. But powder has its own problems from the technology viewpoint. People are crushing blocks and making powder. So we have come up with our own technology or process by which we are making powder directly from sugarcane, concentrated syrup rather. And we have patented process on this, and we are now making powder, mechanize the powder making. Boiler is also installed in the existing plant. With that, we are able to kind of operate multiple effective evaporators and so on. So this is the picture of the plant. Of course, this is dismantled now and shifted to Kolhapur. This was in Varnanagar. And it has a capacity close to 1 ton per day. So this is a demonstration plant. Somebody may ask me a question like what will the cost of this plant and all. As I said, like it's a demonstration plant. We have worked out economics. It won't work at 1 ton per day capacity. It will work at about 5 to 10 tons per day capacity of jaggery production. That means 10 times crushing. Next product is a mobile plant. I have a clip, video clip on this, but in the interest of time, not showing it. Advantage here is, as I said before, we are actually taking the plant to the farm and making powder out of jaggery. And we already sold one unit like in a district called Mandya in Karnataka. We are getting some useful data from them. Last season they operated it, and of course the capacity is small because it's a small unit, but then innovation like this micro reactor and all, where we are actually making it compact, will really help. So we'd like to talk to you and probably see a possibility of integrating your innovation with this compact unit, okay, mobile plant, and many other advantages rather. Your plant goes to the farm and you are getting fresh cane, okay, and fresh cane, making jaggery from fresh cane is something quite beneficial in terms of the quality of the product that we make, in terms of the process parameters that we set. Because even if 2-3 days older harvested cane is it sometimes can give you problems in making jaggery powder, especially jaggery powder. Blocks are relatively easy to make. I was talking about the intervention in the existing units. Now existing units are facing problems, especially from the labor viewpoint, and after concentrating sugarcane juice, juice has to be converted to powder, and right now the way they are doing it's very rudimentary and somehow needs a lot of interventions. Instead of that, we thought of a model where we transport the concentrated sugarcane juice from the existing jaggery unit, bring them to a central unit wherein we actually make powder from it, from our own technology. So this is something that we already tried. Of course, there are still some parts to be proved, but overall it works very well. And we are able to make that jaggery and sell it. So the only thing is the transportation time, which is quite crucial, but the concentrated and clarified sugarcane juice has a longer shelf life than the just crushed cane, whatever raw cane juice that we get. So now what is happening is like we have a project which is a multi-institute project sponsored by RGSTC. IIT Bombay is coordinating it. But there are 4 institutes apart from IIT Bombay, the 3 other, ICT Mumbai, then Shivaji University, and we have RSJR, that is MPKV's arm in Kolhapur on Jaggery Research Station. So in this, every institute is working on different aspects of jaggery making. It's not just the process, but even the product as well. And one of the things that we are doing is making value-added products from jaggery, wherein we have come up with cookies, cupcakes, then tea premix and so on, and not just recipes, but we have actually done profiling and done sensory evaluation, and even we are moving ahead with some nutritional analysis. We have a group working on looking at nutrition aspects of jaggery, because Ayurveda and many other places it's mentioned that jaggery is more nutritious than sugar because of the micronutrients present. So we are actually trying to create scientific evidence by doing in vivo and in vitro analysis, and we are at some advanced stage. We have got some interesting results in terms of glycemic index and so on. So that's the project which is ongoing, but at present in terms of what we can give to market is the plant for making jaggery at a larger scale, about 25 TCD, and of course a mobile plant which is a downscaling of the existing plant. It's about 200 to 300 kilos a day. Overall economics, if you see, like it all depends on, because jaggery price is not fixed, like it's not like a commodity chemical or something. Jaggery price depends on how you sell it, and there are markets, you have to tap those markets, and sometimes you can sell jaggery at 100 or 120 rupees a kilo as well, okay. And there are markets, somehow if the market is ensured, like we are able to give the entire report, economics of making jaggery, both on small scale and large scale. So that's it. Thanks.
What is the land required for that?
Sugarcane is connected from the farm and transported to that point.
Yeah.
And the other is mobile, where it is converted to juice, at least juice, if not jaggery, in situ on the farm itself, right?
No, we make jaggery on the farm.
Jaggery on the farm. So what is the influence of the cost of transportation, because it is influenced by petrol price?
Diesel, yeah.
So I would imagine doing it in situ is a lot more affordable and less expensive than stationary.
Yeah, so you save on transportation cost. You save on even to some extent the quality, like you get better price for the product. But the disadvantage is that the capacity goes down. The overall equipment cost is more. So if you ask me the price of this mobile plant, which is close to 25 lakh for about 200 kilos every day. So that is slightly on the capex side, it is not so attractive. But then, as I said, no, like if you are able to make it more compact by doing micro reactors, making it continuous and all, it will be really, it will meet that requirement.
And the plant need not be bought by the farmer, right?
So we can have different business models.
Different models, yeah.
We can give it on rent. Yeah.
Professor Sanjay, is your video on the laptop? Because to me it talks very effectively. So
I have one. We have. I'll just check.
in the meantime, I think the next speaker can.
On a TCO basis, what would be the capex contribution to the cost of jaggery? I mean, even for a smaller unit, it won't be.
No, it won't be that large. So if I say 15% of 25 lakh,
Yeah.
so it's about how much?
25,000, no,
Yeah,
30,000
divided by
or
200.
something.
So about 5 to 10 rupees.
Yeah.
Yeah. So yeah, as I said, no, if you are
It's
able to
just
sell
initial cost is the one,
Yeah.
but so as you said, the business model can take the initial cost out of the equation.
Right. Yes.
The cane price also is very important, raw material.
Where you can get subsidy for this? You can
Yeah.
put it on there.
We are actually trying that. Yes. Yeah.
Some subsidy may come and the
Right.
cost may be.
Yeah.
ऑडियो नाही आहे का?
ऑडिओ
राहू दे असं चालेल.
नाही ठीक आहे. This is the old one actually. So this goes with the generator also. We thought that suppose we don't get electricity supply on the farm, we can have a generator. So that 2 trolleys actually. So we made many modifications in this. Like our heating system has changed now. Like we use external shell and tube heat exchanger, which is normally used in chemical industry for evaporation.
इथं त्यांचा मायक्रो रिएक्टर लावता येईल.
If this works well, I think this particular mobile unit, if you make it more compact, it's better rather, because you have to transport it, right? So and then in a given volume, if you are able to make more jaggery, it will be good, and this continuous units would really help.
Sir, can you target international markets with your machine?
There are not many countries that make jaggery.
So we addict them with our sugar and jaggery.
अभी
South America, like
मिनियापोलिस में टेस्टिंग हो रहा है ना पाउडर जैगरी का.
where?
मिनियापोलिस.
Uh-huh.
शिवराम मूर्ति.
Yes. But they are testing product, not the
product, not the plant.
Yeah.
Product.
At one point we were, foreigners addicted us to coke and tobacco. We are doing a better job by addicting others too.
Sanjay, the tractor itself
itself cannot, on a stationary basis, cannot supply power. I mean, I don't
Yeah,
know,
that also we
generator,
explored, but that comes
you
from
really need
diesel.
to carry generator.
Right. Yeah. Tractor, we can have a, as you rightly said, like, you can have a diesel
I think
based.
the size of your generator is bigger than the size of your
Right. Yeah. So generator, so there, no, like it's like the power requirement is only for the crusher. Rest all evaporation all is happening through our boiler system, right? But yeah, we can do that. We can have a system attached to a tractor which can generate power.
Okay, thank you. We move to the last presentation before lunch. Dr. Madhu Pinjamur from IIT Bombay.
Yeah, good afternoon, everyone. So someone came for a presentation, and then the presenter asked, can I get 10 more minutes because the presentation is interesting. They said, yeah, you can take 10 more minutes, but you can talk to the walls. So I hope that's not the case here. I think I'm standing between you and lunch. So hopefully that's not the case. So what I'm going to present is a complete feed for small and large ruminants. Small is goats and large is cows and buffaloes, based on ozonated cotton stock as roughage. So this is a collaborative effort between among 3 institutes: IIT Bombay, Nagpur Veterinary College, and BIF Pune. I think we have Dr. Manoj Avre from BIF here, and my colleagues Dr. Suhas Zambre and Rajan Mishra here, and of course Professor Narendra Shah was also one of the participants before he retired from IIT. So what I'm showing in this slide is that fodder shortage is a reality. So these are some news items. You see that in recent years, India has experienced about 23% shortage of dry fodder, 11% shortage of green fodder, so on and so forth. From KrishiJagran.com, government calls meet of central and state officials today to chalk out action plan. Government's ambitious plan to create 100 fodder farmers producers organization has remained only on paper. You want to promote startups for Atmanirbhar in fodder, focus needed on fodder shortage in India. These are all from newspaper items. So the question I think we want to probably look at today is, can cotton stock available in large quantities address this shortage, at least partially? So Maharashtra is no different. Maharashtra also faces a shortage of fodder for feeding cattle. Again, these numbers, the deficit, you see the dry fodder is about 14 million tons, green fodder also tens of millions of tons. All this is coming from a newspaper item. So yeah, ozonated cotton stock could reduce this shortage of roughage, at least partially, because cotton stock goes as a roughage, and for complete feed, animals also need concentrates, as we also need concentrates. So some numbers here, roughly 23 million tons of cotton stock is produced every year in India, and large part of this cotton stock is burnt in the fields and after harvesting cotton. A small fraction also goes for making briquettes, which is used as a fuel in small scale industry. Cotton stock is not consumed by the cattle, neither goats nor cows, buffaloes, of course, because it has got large amounts of lignin, and that makes it hard and not digestible. So we worked on a lab scale and developed a method to make this cotton stock digestible by ozonating it, and up to 65% in vitro digestibility was achieved. At that point of time, we talked to NVC in Nagpur and BIF Pune to set up pilot plants, 1 ton per day at Nagpur and then 2 tons per day at BIF Pune, Urli Kanchan, about probably 1 hour from here, to produce complete feed pellets for goats and cows. And Nagpur worked on goats, and BIF Pune worked on cows. And this ozonated cotton stock was combined with concentrates, minerals, etc., to produce a complete feed. So the process to produce a complete feed for animals is so you have a cotton stalk which is shredded, and then you ozonate it in these tanks, and then you the treated cotton stalk you add it with to the other ingredients that make the complete feed: oil seed cake, dal mill waste. Mix them, grind them, and then you pelletize, and ultimately you see at the bottom you make those pelleted feed, and at the bottom left of me you see these goats which are supposed to be choosy. They are eating this complete feed. So this is a 1 ton per day pilot plant at NVC Nagpur. So all the people you are seeing there are the faculty and students at Nagpur Veterinary College who worked on this, and we also have a similar plant set up at BIF Pune, which is on a slightly higher scale. So based on the method that we developed, pilot plants were set up at Nagpur and Pune, and experiments were done on goats at Nagpur, as I said, and cows at Pune, and weight gain and other physiological parameters were monitored for goats, whereas for cows, I think the parameters which were monitored were milk production, and in addition there were other parameters that were recorded. So in brief, goats that were fed with the complete feed showed significant improvement. So the green histograms here are showing the weight gain grams per day for goats for grazing and complete feed. So those goats which were left to, they were just taken outside for grazing, they gain about a little over 30 grams per day, whereas if they are given this complete feed which contains concentrate and also roughage, they gain up more than 100 grams per day. And on this blue bar graph shows the months the goats take to attain full weight. So when they were left to grazing, it takes more than 20 months, roughly, sometimes up to 2 years to attain full weight. I think that is about 25 to 30 kg. Whereas if they are given this complete feed, they attain the same weight approximately in about 9 months. So we are halving the time they attain full weight. And the feed consumed per day, of course, this grazing, this is a rough number, because there are no. This is just a hearsay, so it's about a little over 400 grams per day, whereas complete feed is about a kilogram per day. And So these graphs show the results with cows. Cows were fed with complete feed pellets, and they also showed a marked improvement. So in the conventional field, the milk yield was about 16.2 liters per day, and with complete feed that went to about 17.4 liters, a little over 7% increase in the milk yield. And the percentage fat, about 3.7% for the conventional feed, whereas a complete feed, they go up to 3.85. And then the price of milk also depends on the fat content. It increases by every 0.1%. You add the fat, the milk price goes up. And the SNF of the milk doesn't change between conventional and the complete feed. So based on the experience at NVC Nagpur and BIF Pune, where we have pilot plants to produce 1 ton and 2 tons per day, we have put some numbers here for a commercial plant which could produce about 10 tons per day, and the numbers look attractive. So the total cost is about 1.6 crores, out of which fixed cost 1.1 and raw material is about 50 lakhs. You need about 100 horsepower. I think that number is wrong there, 100 kilowatt. So you need 10 people to work there. So the cost of production comes to about a little over 13 rupees per kg, and this can be sold at roughly 10-20 rupees per kg. And I think the idea of fixing it at 20 rupees per kg is most of the farmers typically pay a little over 20 rupees per kg to feed their animals, goats and cows or buffaloes. So if you calculate the annual profit, and from there the payback period, 2 years is a payback period, seems okay. So what did I tell you? I think we have available roughage in India is insufficient, for most parts of the country, but that includes Maharashtra. Large quantities of cotton stock are available, and this cotton stock could be ozonated to soften it and make it digestible for cattle. And pilot plants that were set up demonstrated to produce a complete feed with cotton stock as roughage, and remarkable effect, good positive effect was shown on goats, and considerable improvement on performance for cows was also recorded. And the economics do suggest that the cost of producing complete feed is less than the current cost that is incurred by farmers for feeding their animals. And the rate of return is fine, and payback period is about 2 years. And also it is the last point here is also important, I think, which is that the complete feed is was acceptable by the farmers. So our size was, I think it was about 30 farmers. Yeah, about 30 farmers who were given this complete feed over a period of 3 months. So they fed their experimental animals, and it was acceptable, of course, to the animals. They ate, and farmers were also happy. And these farmers also indicated improvement in various other aspects other than milk production also. So I would like to thank Tata Center for Technology and Design, TCTD, IIT Bombay, then RGSTC. Then there are several project staff at IIT Bombay, NVC and BIF, all the 3 institutes who worked on this, and finally the administration of the 3 institutes for supporting the activities of this project. Thank you.
Thank you so much. I think we'll break for lunch and we can take the questions offline. So I request all of you to rejoin in this hall at 2:15 sharp, so that we have 6 more presentations to cover. Dr. Lele will go first. Before that, Anand Chodia would like to say a few remarks. He will chair the session, and then we move to the 5 others. Thank you.